and Assalamualaikum. My name is Siti Nursahidah binti Muhammad Azman with the matrix number of 20360. I'm from BMY3201, Basic Technique of Microbiology, Group 1. So today I'll be explaining and demonstrating two experiments, which is tricking for single isolated colonies and also gram staining. For each experiment, I will explain briefly the principle, and then I will demonstrate the method, and at the end I will share the expected result. So without further ado, let's start with the first experiment, which is streaking for single isolated colonies. So basically, this technique was used to gain pure culture. According to Cappuccino and Wells, pure culture is a culture that contains only one type of organism, and it is suitable for the study of their culture, morphological, and also biochemical properties. So in order to gain this culture, as we streak the organism on the agar plate, we need to reduce the number of the organism so that at the end, the cell can be sufficiently far apart. So when discrete and well isolated colonies are gained on the nutrient agar plate, we can easily pick up with the sterile needle or loop and transfer to the wanted media. So when the culture grow on the wanted media, it is known as stock or pure culture. So now let's proceed with the method. Before we begin, let me introduce the materials needed for the experiment. For the culture, we will use a mixed culture. For the media, we will use a nutrient agar plate. And for the equipment, we will use a ruler, marker pen, inoculating loop, Bunsen burner, and also a lighter. As a beginner, I will divide the agar into 4 sections using permanent marker and ruler. I draw a bisecting line on the bottom of the petri dish. This will ensure that I'm not touching the area that have been inoculated, so that I'll get a good result. Then, I number each quadrant in counterclockwise rotation. Next, I label the agar with my name, name of culture, and the date of experiment. For the next step, we will require an inoculating loop, Bunsen burner, lighter, a mixed culture, and a nutrient agar plate. Firstly, I will turn on the flame. Then, I will flame the inoculating loop. The inoculating loop should be flamed until it's red hot. Next, I allow the loop to cool. If we are not sure whether the loop had cooled or not, we can touch it to a blank area of the agar. If it's too hot, it will sizzle and melt the agar. Then, I uncap the bottle of the mixed culture and keep it within my palm. Then, I flame the neck of the bottle and I dip the inoculating loop into the culture. And I reflame the neck of the bottle. After that, I make a primary inoculation at the first quadrant. Make sure to inoculate at the sterile area. Then, I reflame the loop and let it cool. I rotate the plate to the next quadrant and make a second set of streak by touching the corner of the culture in the area 1 and drag it several times across the agar in area 2. Note that the loop should never touch the primary area again. After that, I reflame the loop and allow it to cool. Then, rotate the petri dish and inoculate quadrant 3, as what I have done in quadrant 2. For the quadrant 4, we don't need to reflame the loop. Instead, I just inoculate the quadrant 4 as the previous quadrant. After I finish inoculated, I reflame the loop again to sterilize the loop. Now, let's look to the result that we can expect. We should see the highest growth happen at the first quadrant, and the growth of the culture was reduced at quadrant 2, 3, and 4. And the last quadrant has the smallest amount of growth, which is 3 to 4 small dots, where actually it is a single colonies of an organism. Okay, that's it for the first experiment. Now let's move on to the second experiment, which is gram staining. We all knew that microorganism was so small that it cannot be seen with naked eye. It even transparent and colorless when suspended into aqueous medium. So staining is one of the ways for us to visualize the cell. There are several staining techniques, and each staining technique have its own purposes. In this video, we will look into depth about gram staining. So gram staining was named after Dr. Hans Christian Gram. It was one of the most important differential stain. It will divide the bacterial cell into two major groups, which is gram positive, which will appear in purple, and also gram negative, which will appear in pink when observed under the microscope. This difference can be differentiated as the bacteria have different chemical composition in cell wall. Gram staining requires at least 4 reagents to be applied sequentially to the heat-fixed smear. The first is primary stain, which we use crystal violet, which purple in color, and it will stain all the cell. Next is mordant. Mordant will increase the affinity of the cell for stain by forming an insoluble complex. We use grams iodine as mordant. Third is decolorizing agent, which we use 95% ethyl alcohol. It may or may not decolorize the primary stain. Note that we cannot over decolorize, and it will remove all the primary stain. And lastly, we use safranin as counter stain. Counter stain will stain back all the cells that have been decolorized. So next, let's proceed with the methods. Before we begin, let me introduce the materials needed for this experiment. The culture needed is a broth culture of Escherichia coli and a broth culture of Staphylococcus aureus. The reagent used is crystal violet, grams iodine, 95% of ethyl alcohol, and safranin. And the equipment will be used is a Bunsen burner, inoculating loop, staining tray, glass slide, and bibulous paper. Next, for the method, after the bacterial smear have been prepared, gently flood smear with crystal violet and let it stand for 1 minute. Next, gently flood smear with grams iodine, mordant, and let stand for 1 minute. Then, gently wash with tap water. After that, decolorize with 95% ethyl alcohol. Note that do not over decolorize. Add reagent drop by drop until the alcohol runs almost clear, showing only a blue tint. Once again, gently wash with tap water. Lastly, counter stain with safranin for 45 seconds. And then, once again, gently wash with tap water. Then, you can blow dry with bubbleless paper and examine under oil immersion. Lastly, for the expected result, when we observe the smear under the microscope, for the broad culture of E. coli, we can see a bacilli shape and it arranged in cluster. The cell also will appear in pink. Next, for the Staphylococcus aureus, we can see it is cocci in shape and also in cluster arrangement, but it is in purple color. Okay, that's all from me. Thank you.